Hello and welcome to today's video lesson. It is on neurohomeostatic control pathways. And we've been talking about homeostasis. We said last week that the word homeostasis, it's from the Greek words that mean same and steady, and it refers to any process that living things use to actively maintain fairly stable conditions that are necessary for survival. So today we're looking at how the central nervous system, how electrical impulses, messages sent throughout our body play a pivotal role in homeostasis. So the learning goals for today are, let's get into it. First up, to identify that these cells that transport nerve impulses throughout our body. We'll look at the structure and function of the neuron. We will also discriminate between different types of neurons. So there's sensory and motor. There's also interneurons, which we'll get to. Then we will explain the process of how nerve impulses travel. So firstly, within the neuron, that's called an action potential, how it moves, but then also how that impulse travels between neurons. So we're going to look at synaptic transmission. So you can pause the video here and write just some kind of summarized version of those goals. Cool beans, let's get into it. So first up, cells that transport nerve impulses. I'll start off by sharing some interesting facts with you. The brain is amazing. It has about 100 billion neurons. That is as many, roughly, stars as there are in the Milky Way galaxy. As many stars in the galaxy that we live in, that's how many neurons are in our brain. The brain is amazing. It is constantly changing in response to our environment, in response to our experiences, and that's called neuroplasticity. Not only is it constantly changing, but it's constantly producing new neurons. And this process is called not neuroplasticity, but neurogenesis. Genesis meaning like beginning, new. Did you know that signals in your brain, in your neurons, can travel at the same speed as Formula 1 cars? That's up to about 360 kilometers an hour. Have you ever been that fast? I think when I was skydiving, I got up to close to 200 kilometers an hour in freefall. Pretty awesome. So, neurons, what are they? Here's an image. They are the fundamental units that send and receive signals which allow us to move our muscles, feel the external world, think, form memories, and so much more. They are specialized cells. That means they've become specialized through that what was once stem cells. Now they are performing a particular function and they look a certain way. They are specialized to transmit electrical impulses within the nervous system. The nervous system converts what we take in from our senses, that's called sensory information, into these electrical impulses in order to make sense of our environment, to rapidly detect, but also to respond to stimuli. And while there's different types of neurons in our brain, it's quite complicated how we categorize all these different types. There's three basic components to all the different neurons that we know. So if we look at this image here, we have these branches where it spreads out, and these are called dendrites. They're short branched fibers, and they convert chemical information from nearby neurons into electrical impulses to travel throughout the neuron. Then we also have this kind of spinal looking shape, and it's this elongated, that means like stretched out fiber that transmits these signals to the terminals, where they can then pass on to the next neuron or the next part of the system. This is called the axon, and it's coated in this, it's wrapped up in this like fatty-like substance, and that's called myelin sheath. That has a very important role, which we will talk about later. Then we also have the soma. So that's like this body, and that can change in location depending on the type of neuron, but they all have a soma with a nucleus. So that's the body. And as we said, this myelin sheath, basically that improves the conduction of speed of electrical impulses. But it also requires additional space and energy. So we have this where we need it, but not every neuron has this lovely myelin sheath. So that's first up. Then we're going to talk about now different types of neurons. And if you just look at a neuron through a microscope, it's clear that not all neurons are the same. So how many types of neurons are there? And how do scientists decide on these categories? Well, in the brain, that's not an easy question to answer. For the spinal cord, though, we can say that there are 3 types of neurons. And that is sensory, motor, and interneurons. But if we just focus on those first 2, if we talk about sensory and motor, you can see that just looking at it initially, they have a different structure. That's because they have a different function as well. We don't understand everything about how structure relates to function, but we can identify or discriminate a few features between them. So let's talk about sensory neurons to start with. We can identify dendrites, we can identify axon, and we can identify the body, the soma. You can do the same thing here on the motor neuron, and you can see that, again, it's different. Let's just say what they do. Sensory neurons carry sensory impulses from sensory organs, our eyes or our fingertips or whatever other senses you want to talk about, to the central nervous system. And so they're called sensory neurons, as opposed to motor neurons, which go the opposite direction. They carry, now that the brain has perceived and made sense of that sensory signal, now it needs to go back to the effectors, the muscles or the glands. So that's what motor neurons do. They carry motor impulses from the CNS to specific effectors, motor neurons. So if we talk about a few of their differences, I'll talk about interneurons again in a sec. We can distinguish between the main features. So in the soma, you can identify that in sensory neurons, it's this bulge on the side of the axon. We don't fully understand why it's there, but we think, well, if sensory neurons pick up info and send it to our brain, that needs to happen quickly. Maybe it's something to do with speed. As opposed to the soma, which is in motor neurons, it's in the dendritic end of the neuron. We can look at the dendritic terminals and see it's a little bit different. In the motor neuron, the soma branches out into fine thread-like extensions. They have synapses, which receive signals from other interneurons. However, in sensory neurons, the dendrites branch off one end of the axon instead of the soma. So you can see that there. Axon terminals. At the opposite end to the dendrites are the axon terminals. In sensory neurons, the axon terminates in many synapses that link and send signals to interneurons. While in motor neurons, the axon terminates in structures called end plates that interface with muscles or glands directly. So these could be hooked into different sections of a gland or onto the muscles, because remember, they're creating an effect. Direction of impulse. While the impulses in sensory and motor neurons both travel from dendrites to axon terminals, the direction of the impulse in relation to the body is different. And you can see that here. So if the CNS is here, we go this way, but then if we're going back from dendrites, CNS, dendrites, we're going out to the effectors. So to the CNS, from the CNS. Cool. So lastly, interneurons. They connect various neurons within the brain and spinal cord. So they have this, they're not necessarily going from the senses to the CNS or from the CNS to the effectors, but they're in that in-between range of getting that message passed up from the spine to the brain and from the brain to the spine and then back out again. That is why they are called interneurons. I'll speak about nodes of Ranvier shortly. Let's talk about how messages actually move though through a neuron. So there's a few phases to this. But before we get into the impulse actually traveling, I'll talk about what is going on when it's not traveling. And this is called resting potential. And basically, if I bring up a diagram, I'll talk through this. Neurons generate and conduct electrical signals by pumping positively charged ions, so that's like sodium and potassium, across their membrane. So here we have like the outside, and here we have the inside of the neuron, the axon. It is the unequal distribution of these ions on different sides of the membrane, here's the membrane in gray, that creates a membrane potential, this difference in charge. A resting potential is the difference in charge across the membrane when the neuron is not firing. It's more negative inside the neuron relative to the outside. So it's actually not a negative charge, but compared to the outside, it's quite negative. And the way that this happens is because of the sodium-potassium pump. It's this pump that does active transport in it. And what it does is it puts out 3 potassium ions for every, 3 sodium ions, I should say, as you can see in this diagram, 3 sodium ions for every 2 potassium ions. And what that's doing, it's building up this charge, and you can see that there's heaps of potassium outside, and heaps of sodium outside and heaps of potassium inside. And now there's this difference, and the inside, we could say, is more negatively charged. So there's this movement. What happens is there's this potential for something to happen because of resting potential, because of the sodium-potassium exchange pump. So that's what's happening when there's no message firing. Let's talk about when there is a message firing. So that is called the action potential. And this is how a nerve impulse moves throughout a neuron. Now there's 3 main stages to this. The first stage, well, the 3 stages are depolarization, repolarization, and a refractory period. So let's talk about the first one, depolarization. It refers to a sudden change in that potential that I was just talking about, from a relatively negative environment on the inside to now a very positive change. And what happens, it's in response to this sodium channel that opens. So all that sodium outside, now that there's this change happening, the sodium channel opens, and just passively, where's all that sodium going to want to go? From an area of high concentration to an area of low concentration. So this is called the influx of sodium, and it causes the membrane potential to become more positive on the inside, and that's called depolarization. The next stage is, if you look at this diagram now that sodium channel is closed, but that has now caused this next channel to open up. That's the potassium channel. It opens, and so what's going to happen? Well, all that potassium on the inside is now going to move to the outside. So this is called the efflux of potassium, and it causes the membrane potential to return to a more negative internal differential. Basically, if you just know the process of, okay, first step, all this comes in, sodium comes in, and then the next step, that triggers all the potassium to go out. So that's repolarization. And then what happens next? We have the refractory period. And this refers to the period of time following a nerve impulse where potassium ions are mainly inside and sodium ions are mainly outside. And it's this state of kind of returning back to the resting potential. It's where the ion distribution is being restored. So if I have an animation, which I do shortly coming up, it'll go through this with you. But if you just kind of know the main steps, depolarization, it's an electrical charge within a neuron from a relatively negative charge to a positive charge. And that's because all this comes in, then that stimulates a relatively positive charge to a negative charge. Now that goes out, and now we have this returning back to that resting potential with the sodium-potassium exchange pump. Now going back to that initial state of pumping the sodium out and of pumping the potassium back in. Next step. So that's how an impulse travels through inside a neuron. What happens when it gets to the end, to those dendrites? What's going to happen? So this is all the synapse I'm in. This is called synaptic transmission. And basically, there's a few steps that happen in synaptic transmission. Back in 1963, I will say firstly, though, that a prize was awarded for work done to understand how these action potentials work. And we can see that there's a few steps to this process. So I'll just talk through this image with you to keep it simple. One, now we've got that action potential that has come down. What that does is it causes these calcium channels to open. And so what that allows is calcium to enter the presynaptic neuron. So if here's the synapse, pre means just before. So calcium enters this neuron, and then it's put into these like little vehicles, or what we call vesicles. So that means it's about to bind to the membrane so that it can then be released. So the vesicles move to the membrane and dock, and then they release this. It's called a neurotransmitter. It's this chemical that's going to stimulate an actual potential in the receiving neuron. And this happens via exo, that means out, cytosis. So now these neurotransmitters bind to these receptors, and that signal is now initiated, and that's going to cause that whole process that we just looked at before in this new step. And so that process I'm talking about is depolarization, repolarization, and then the refractory period. So this region is called the synapse. This is the presynaptic part, and then this would be the postsynaptic part at the end. And that's how we get messages between neurons, or passed on to other neurons. So let's have a look at what this might look like. And it's also comparing myelinated to unmyelinated. So one's going to be a lot faster. Which one's that? It's the one that has the myelin sheath wrapped around it. So it's just kind of showing how a nerve impulse travels through, and myelin sheath makes it happen a lot quicker. One other thing I wanted to point out was just this section of the axon where you can see myelin sheath is wrapped around it, but what's those sections in between? We called those nodes of Ranvier. And that is all for this week. Hope that you have been able to take a little bit from this. So your homework, as always, is just to check out chapter 10 of the textbook and add those keywords to your glossary. Guys, I hope that you have found this video informative and interesting and helpful leading up to all your study and your final exam. I hope that you can come back to this throughout the year and just have a little refresher on everything that we've done this week. God bless you guys. I'll see you in class.